Welcome back. This is Biology 2210 Lab, Anatomy and Physiology 1 Lab at CNM. This week's topic is the brain, and this is Chapter 14 in your textbooks. So, of course, when we talk about the brain, we're talking about the other part of the central nervous system. We talked about the spinal cord previously. The brain, of course, is located in the skull and weighs about 3.5 pounds or so, a little bit less in women than in men. And it really is the major center for many of the nervous system's functions. In terms of overall organization, there are 3 parts to the brain. These 3 parts are the cerebrum, the cerebellum, and the brainstem. The cerebrum is the part of the brain that most people think of when they think of the brain. It is about 80% of the brain's total volume, and it consists of all those lumps and folds that we think of underneath the skull when we think of the brain. The cerebellum is a posterior portion of the brain, sitting just behind and below the cerebrum. And even though it's not a big percent of the brain by volume, it does contain, as you see here, 50% of the brain's neurons. So it's a very active region of the brain. And then finally, the brainstem is a stalk-like structure that we find at the center of all of this, the cerebrum and the cerebellum. And this stalk-like brainstem is connected ultimately to the spinal cord. And so most of the messages coming from the spinal nerves through the spinal cord come up through the brainstem. Of course, remember, we do have cranial nerves that allow some messages to pass to and from the brain and bypass the spinal cord and the brainstem. All right, if we look at the surface of a brain, again, we can see some of these structures that we talked about. Again, the cerebrum is all of this area here with all the lumps and folds. That is, again, the part that most people think of when they think of a brain. The cerebellum is, as I said earlier, posterior and dorsal or behind the cerebrum, the second largest part of the brain. And then the brainstem, again, this stalk-like structure from which the other structures seem to emanate. The brainstem will continue up higher up into this region as well underneath the cerebrum. And here we see the spinal cord, of course, merging and becoming the brainstem as it travels up through the foramen magnum of the skull. While we're on this view, I just want to point out again that up in the cerebrum, all these lumps of tissue, all these folds are referred to as gyri. Each one is a gyrus. And so the surface of the cerebrum has all these lumpy-like folds, twisting folds, each one referred to as a gyrus. The depressions or the invaginations between the gyri are referred to as sulci, and singular for sulci is sulcus. And if you were taking a class in neuroanatomy, you would learn some of the names, from the very specific names of each gyrus and sulcus. In this particular class, we're not going to ask you to learn the names of particular gyri and sulci, except for just a few. For instance, there is a very clear sulcus that is kind of located halfway back on the cerebrum. This one right here. This is known as the central sulcus. This is an important barrier, as we'll see later, between what's called the frontal lobe and the parietal lobe. There's another pretty obvious sulcus right here called the lateral sulcus. This lateral sulcus, again, a fairly obvious, easy to identify sulcus, and it also is a border between, again, the frontal and parietal lobes above and what we call the temporal lobe down here. Towards the end of today's presentation, we will talk about these various lobes of the cerebrum. Of course, if sulci have names, then the gyri, again, have names as well. Once again, we're not going to ask you to learn any except for 2, what we call the precentral and postcentral gyri. Those have to do with this central sulcus. So this gyrus right here, just in front of the central sulcus, is called the precentral gyrus, and that's going to be an area where we have lots of motor control, sending messages out to skeletal muscles of the body. And then this one, just behind the central sulcus, is the postcentral gyrus. Postcentral gyrus. And this one is now, of course, part of the parietal lobe, because the central sulcus, again, is the boundary between the two lobes. And this one, this postcentral gyrus, serves a lot of sensory function, a lot of touch sensation from the body. But again, we're going to talk more about the lobes and the functions of the lobes towards the end of today's presentation. We also have fissures in the brain, or in the cerebral or parts of the brain. Fissures are like sulci, except they're deeper, much deeper than sulci. So, for example, there's a very deep fissure here between the cerebrum and the cerebellum, much deeper than a typical sulcus. This particular fissure happens to be called the transverse fissure. If we changed view and looked at this brain from the top, we'd also notice that the cerebrum is broken up into two hemispheres, two halves, two what we call cerebral hemispheres. And the fissure that splits the cerebrum up into two hemispheres is called the longitudinal fissure. So you have a lateral fissure on the side and then a longitudinal fissure down front and center. And here again we see the three major portions of the brain that we talked about: the cerebrum, the cerebellum, and the stalk-like brainstem. Again, we've already talked about the gyri and sulci and fissures that are very characteristic of in terms of surface anatomy. As I said earlier, if you looked at the brain from the top, you'd clearly see that the cerebrum is organized into two hemispheres, and here's the, again, longitudinal fissure that I was referring to earlier. Now, one thing I want to mention at this point about today's video is that the PowerPoint in your book does not follow the sequence of the list of terms, and that's just a difference of opinion in terms of how best to proceed. Normally, if this was face-to-face, I would jump around in the PowerPoint, but given that we're recording, I don't want to do that. And so I'm going to follow the sequence in the PowerPoint, which means we're going to be jumping around a little bit in the list of terms. So we will get to every term, but you want to pay close attention as we go, because you'll have to be jumping around a little bit in your list of terms to follow the sequence of ideas in this video presentation. For those of you that have the PDF of the PowerPoint, you'll also notice that I have eliminated a lot of the slides that I would normally just run through, and that just makes things easier as well. I've also rearranged some of the sequence just to make the ideas flow. So unfortunately for today, the sequence of terms in your list of terms is not going to match exactly the sequence here, and your PDF that you have of this PowerPoint won't exactly match either. But I hope that won't present too great a problem. Alright, here's another view of a photograph of a brain showing you once again sulci, gyri, and the longitudinal fissure. Another photograph highlighting what gyri are versus what sulci are. And if you travel down in the longitudinal fissure far enough between the two hemispheres of the cerebrum, what you're going to find is eventually you find a place where the two hemispheres join together. So there is a connection between the two hemispheres of the cerebrum. So again, as I said, if you travel down the longitudinal fissure far enough, eventually you'll hit this structure. This structure is known as the corpus callosum. The corpus callosum consists of tracts of white matter that are connecting the left hemisphere to the right hemisphere. In a sagittal section, the corpus callosum looks like a sort of a comma-shaped structure connecting the two sides. Now, I referred to this corpus callosum as tracts of white matter I need to define a couple of things here. First, what is white matter, and second, what is a tract? So you'll notice in this frontal section of the brain, if you look carefully, you'll see that there's clearly two distinct kinds of tissues in this photograph. There's a darker tissue and a essentially white creamy colored tissue. The darker tissue is referred to as gray matter. The lighter colored creamy tissue is called white matter. And there is a functional and structural difference between gray matter and white matter. What you have when you see gray matter is the cell bodies of neurons. This is where all the neuron cell bodies are located, is in gray matter. It also consists of axons, but in gray matter the axons are unmyelinated axons. So you have neuron cell bodies, dendrites, of course always associated with neuron cell bodies, and unmyelinated axons. The white matter, on the other hand, consists of myelinated axons. So in terms of brain power, in terms of processing power, it's the number of neurons that counts. And so organisms that have more gray matter are going to have brains that are capable of more processing. White matter really, again, just consists of myelinated axons. So white matter just consists of fibers that are making connections, sending action potentials to and from different parts of the brain, or perhaps from the brain down to the brainstem and then out to the body. All right, and a tract, basically a tract is a bundle of axons. So what we call a bundle of axons out in the body, in the peripheral nervous systems, we call that a nerve when it was out in the body. But in the brain, when we have organized bundles of axons, we call them tracts. Very similar to nerves, but not so physically distinct from surrounding tissue. And so we use tracts to distinguish them from nerves. And so we said the corpus callosum is a tract of white matter, tracts of white matter, again making connections, sending messages back and forth so the two hemispheres of the cerebrum can talk to each other. As we'll see later in the presentation, the two hemispheres are not completely identical in their function. They tend to complement one another, and for total brain function to work, we have to be able to communicate one side to the other to get that full complement of brain function. So the corpus callosum is a very important part of the brain, allowing communication between right side and left side. A severe treatment for epilepsy in the past used to be severing of the corpus callosum. For some reason, that reduced the severity of epileptic seizures. Of course, by severing the corpus callosum, that prevents then the two hemispheres from talking to each other, and it creates some cognitive problems for individuals. One other thing I wanted to mention about gray matter is you'll notice that in the cerebrum, much of the gray matter is located at the surface. Excuse me. And as I said earlier, brain power, processing power, is really based on how much gray matter you have. So this arrangement allows us to understand why the surface of the cerebrum is folded so much into gyri and sulci. The reason it's folded is to increase surface area. Because where is most of the gray matter? At the surface. So the greater the surface area, by folding, the more neuron cell bodies we can have, and therefore the more powerful, the more processing power we can have in the cerebrum. You'll notice that gray matter isn't restricted to the surface. We also have some islands of gray matter that seem to be embedded in the white matter. Here we see a nice little patch of gray matter embedded in white matter, and we see some here as well. These are generally referred to as nuclei. Patches of gray matter embedded in white matter referred to as nuclei. And again, this consists primarily of neuron cell bodies and unmyelinated axons. Nuclei are kind of similar or analogous to ganglia in the peripheral nervous system. Remember, in the peripheral nervous system, you do find occasionally neuron cell bodies out there, and they're in clusters called ganglia. In the brain, we have clusters or islands of neuron cell bodies, but they're called, again, nuclei. So here again is just a review of some of the things we've already talked about. Again, the cerebellum is the second largest part of the brain. It's only about 10% of its volume, but 50% of the neurons, as we said previously, are in the cerebellum. Then, as we said again, the brainstem is what remains if you take away the cerebrum and the cerebellum. It's the stalk-like center of the brain. And at this point, I want to just point out that the brainstem itself is generally divided up into four major regions, working from top down. Those are the diencephalon, the midbrain, the pons, and the medulla oblongata, often just referred to as the medulla. And again, during this presentation, we will talk about each one of these regions of the brainstem in sequence. Again, just a review of the surface anatomy of the brain that we've talked about. And again, here are the portions of the brainstem that we just mentioned. As we said earlier, working from top down, we have a region kind of deeply embedded at the center of the cerebrum called the diencephalon, a small region just below that called the midbrain, and a lump of tissue known as the pons, and then the inferior most part of the brainstem, the medulla oblongata, which fuses or merges and transitions into the spinal cord. Here's a view, again, a sagittal section of the brain, and a couple of things to point out here. There's the corpus callosum again, making connections from left side to right side. There's your cerebellum. One thing I want to point out in the cerebellum is that all the extensive gray matter you see in the cerebellum, very little white matter, mostly gray matter, and that's why we have such a high percentage of neurons of the brain located in the cerebellum. All of this is the brainstem, and essentially we have the diencephalon, which is this region up here, the midbrain, small region right in here, the pons, as I said, a lumpy structure in the anterior part of the brainstem, and then another swollen region, the medulla oblongata. And a picture, an actual photograph of what we just looked at. And again, a summary of gray matter versus white matter, which I've already gone through. Here again, we see the distribution of gray matter versus white matter. And once again, just because it's highlighted, you see much of the gray matter at the surface of the cerebrum, and then patches of gray matter, nuclei. Okay, at this point, we alter, again, as I said earlier, the sequence a little bit, so we have to jump ahead in the list of terms to where we begin to talk about the meninges. Now, we are familiar with meninges because we talked about the meninges in our discussion of the spinal cord. And so you already know that around the spinal cord there are three meninges, a tough dura mater on the outside, an intermediate arachnoid mater, and a pia mater, a softer pia mater right on the surface of the spinal cord. Well, we have the same three meninges in the brain, surrounding the brain, and they are in the same sequence, serve the same functions, have the same basic features. The one obvious difference between the meninges of the brain and the meninges of the spinal cord is that, recall in the spinal cord we had something called an epidural space. There was a fat-filled space between the dura mater and the bone of the vertebrae. And we mentioned this is where, for instance, epidural injections are made if you want to anesthetize the lower extremities, say during labor and delivery. We don't have that space, that epidural space here in the skull. In the skull, the dura mater adheres directly to the bone, the cranial bones of the skull. So there is no space between the dura mater and the bones of the skull. There is no epidural space. So if you, this was best seen when we used to have cadavers, and you could see that the dura mater, the very, very tough protective dura mater, would literally have to be ripped off the inside of the skull to free it up and expose it. Another interesting thing about the cranial dura mater is that it does have two layers. An outer layer, again, that's connected to the bone of the skull, called the periosteal layer, and then an inner layer called the meningeal layer, which is more typical of dura mater. The fact that it has two layers can be seen best in places where those two layers separate. And those two layers separate at the center of the brain, where the dura mater dives down between the two hemispheres. So remember, there's a longitudinal fissure between the two hemispheres. A portion of the dura mater will actually extend down into that longitudinal fissure. And this portion of the dura mater that extends down and separates left from right hemisphere is referred to as the falx cerebri. The falx cerebri. This helps, again, protect the two hemispheres of the brain, but it also serves as an anchor point, because the falx cerebri at the anterior end, you really can't see this here, but at the anterior end, the falx cerebri attaches to the crista galli of the ethmoid bone. So you may remember back to your skull structure earlier this semester, a little ridge of bone sticking up off the floor of the cranium. So on either side we had the cribriform plate, but that ridge of bone was called the crista galli. Well, this is what the falx cerebri attaches to at the front end, and so this is an anchor point, and this helps the dura mater then stay anchored, which of course then helps stabilize the cerebral hemispheres, which are inside of it. On the back side, the falx cerebri spreads out again and becomes something called the tentorium cerebelli, which is not on our list of terms. It is on our list of terms. The tentorium cerebelli. And the tentorium cerebelli essentially helps separate the cerebellum from the cerebrum. And the tentorium cerebelli also anchors to the floor of the cranium. So you have an anterior anchor point, a posterior anchor point, and this is basically important in helping to hold everything together and stabilize the movements of the brain and cerebellum. Arachnoid mater and pia mater are similar to our description of them in the spinal cord. So I'm not going to say too much other than to remind you that the subarachnoid space with the cobwebby-like extensions does contain cerebrospinal fluid. This cerebrospinal fluid is important in, again, serving a shock absorption function and helping the brain float within the cranium. And so, again, it serves a cushioning and a buoying type function within the skull. I mentioned earlier that the two layers of the dura mater do separate near the center, the sagittal line of the skull. And here we can see a nice view of that. This is just a sagittal section, a frontal section through a portion of the brain. Right, so a frontal section cutting this way. We imagine we've taken a wedge and pulled it out and are just looking at that wedge-shaped piece that's been pulled out. The dura mater, again, the outermost Meninges here, arachnoid mater underneath it. There is a subdural space between these two. Your little cobweb extensions of the arachnoid mater into the subarachnoid space. This is filled with, again, cerebrospinal fluid, cushioning the brain. And then a pia mater would be right on the surface of the brain. We mentioned earlier there's two layers to the dura mater, an outer periosteal layer which is attached directly to the cranial bones, so no epidural space, and then the meningeal layer. And I said, again, along the sagittal line, right along this central sagittal line underneath the cranial bones, you can see that the two layers separate. And so there is a little fluid-filled space here that runs the length of the dura mater underneath, again, this mid-sagittal line. This space is referred to as the superior sagittal sinus. Now, sinus is a word you're going to learn in A&P2. A sinus is actually a kind of blood vessel. Specifically, a sinus is a type of vein usually involved in collection of fluid. And so that's what we're seeing here, is filling this space between the two layers of the dura mater, running the length of the mid-sagittal line, is a large, irregularly shaped vein called the superior sagittal sinus. And this vein is collecting something specific. It is collecting cerebrospinal fluid. So this superior sagittal sinus is a drainage site for cerebrospinal fluid. So at this point, we want to talk a little bit about cerebrospinal fluid. It is produced by capillaries in the brain. And let's see if we can take a look at the next slide. Hopefully the sequence will show us this. We have a couple of slides on meningitis that I left in here, just FYI. It's not on our list of terms, but I left them in just thinking perhaps some of you might just want to take a look at what meningitis is. Okay, so back to the cerebrospinal fluid. As I said, cerebrospinal fluid is produced by capillary beds, and these capillary beds are referred to as choroid plexuses. So a choroid plexus is a special kind of capillary bed in the brain excuse me, that leaks out cerebrospinal fluid. And so we see cerebrospinal fluid really is a derivative of blood plasma. It's not a secretion. It's not a cellular secretion. It really comes from blood plasma that leaks out of these capillaries called choroid plexuses. Slightly modified because the brain is very sensitive to chemicals. The brain is largely a chemical organ with all the neurotransmitters at its synapses, and we don't want any old chemicals just leaking out into the brain tissue. So we are pretty careful about what we allow to leak out and become cerebrospinal fluid. But the interesting thing to note about this origin of cerebrospinal fluid is, one, it's constantly leaking out of these capillary beds, these choroid plexuses. And so it's not static. And because it constantly is leaking out, it must also constantly be drained away. Otherwise, it would accumulate inside the brain and it would cause swelling and damage. So cerebrospinal fluid is not static, it flows. Its origin is in the choroid plexuses, and it drains away into that mid-sagittal sinus, that sagittal sinus, that vein that we saw runs, again, just underneath the skull along the sagittal line. So we know where it begins in these choroid plexuses, and we know where it drains back into the bloodstream, because remember, the sagittal sinus is a vein. The question is, how does it flow? How does it get from its origin in the choroid plexuses to the sagittal sinus? Well, we're going to answer that question in just a minute. The other thing I want to point out about cerebrospinal fluid, however, before we talk about its flow, is that because it derives from the blood plasma, it does contain some important materials that are in the plasma, such as oxygen and nutrients. And so the cerebrospinal fluid does supply oxygen and some nutrients to the tissues that it's in contact with, the tissues that it bathes. And like blood plasma, it can also pick up carbon dioxide and waste products and drain those away. So this flow of cerebrospinal fluid is important, not just to make sure that cerebrospinal fluid is distributed around the brain and spinal cord for support purposes and cushioning, but also because of the metabolic support that the cerebrospinal fluid provides. Okay, so back to the flow. where are these choroid plexuses located in the brain? Well, choroid plexuses are located in specific hollow chambers in the brain known as ventricles. So the brain, the cerebrum, and the brainstem are not solid all the way through. There are these hollow fluid-filled chambers within the cerebrum and the brainstem, and these are referred to as ventricles. We have 4 ventricles in the brain, and the first 2 are up in the cerebrum, and they are referred to as the lateral ventricles. So we have a right lateral ventricle and a left lateral ventricle, best seen, of course, in this anterior view. A right lateral ventricle and a left lateral ventricle. These are the first 2 ventricles. It's difficult to describe their shape, I guess, kind of C-shaped in a way, but they do have some projections that make it not a perfect C. But interesting C-shaped, basically C-shaped hollow chambers in the cerebral hemispheres. Each ventricle has a choroid plexus associated with it, producing cerebrospinal fluid. Now that cerebrospinal fluid is not trapped in the ventricle, of course. As we said, we have to eventually get it to the sagittal sinus at the top of the brain. And so what happens is cerebrospinal fluid that's produced in these lateral ventricles can move out of the lateral ventricles through little channels called interventricular foramina. Each one is an interventricular foramen, and they can pass through to a third ventricle, which is located at the center of the diencephalon or brainstem. So this little thin chamber here, from the side we see it shown here, this is known as the third ventricle, and it receives cerebrospinal fluid from the first 2 ventricles, the lateral ventricles. There is another choroid plexus in the third ventricle, so it also adds its own cerebrospinal fluid to what it's receiving. And from the third ventricle, all the cerebrospinal fluid will eventually exit through a channel known as the cerebral aqueduct. The cerebral aqueduct. It's a channel that carries cerebrospinal fluid away from the third ventricle. So again, this is the third ventricle up here receiving from the lateral ventricles, and then the cerebral aqueduct, an extension of the third ventricle, which carries the fluid down to the 4th and final ventricle, which is simply known as the 4th ventricle. And you see the 4th ventricle is fairly far down on the brainstem, just behind the pons and in front of the cerebellum. Once again, this 4th ventricle has its own choroid plexus, so it adds some cerebrospinal fluid to what it's receiving from the other 3. At this point, the cerebrospinal fluid has some alternatives. A small amount of the cerebrospinal fluid will pass down into the spinal cord via the central canal. So remember that when you looked at a section, transverse section through the spinal cord, it had a central canal right at its center. That central canal is filled with the cerebrospinal fluid. This cerebrospinal fluid will pass all the way down through the spinal cord, and at the end of the spinal cord, at the solid tip, will exit the spinal cord and enter the subarachnoid space. And then it will gradually work its way back up in the subarachnoid space. Most of the cerebrospinal fluid, however, will exit the 4th ventricle through these openings that we see here. There are 3 openings by which the cerebrospinal fluid can exit the 4th ventricle. 2 of them are referred to as lateral apertures, and of course they are, as the name implies, lateral, sending cerebrospinal fluid out to the side. And then we have a single median aperture that's located right at the center of the 4th ventricle. In all 3 cases, the cerebrospinal fluid is again exiting into the subarachnoid space of the meninges. So all the cerebrospinal fluid that's been produced in these ventricles eventually ends up in the subarachnoid space, either of the brain or the subarachnoid space of the spinal cord. But again, the goal is to ultimately drain this cerebrospinal fluid into the sagittal sinus, that vein embedded in the dura mater on top of the skull. So let's see what happens. This is a photograph of a cross-section through the brain. And again, you see gray matter and white matter nicely here, but mostly I'm letting you see this because I want you to see these hollow chambers. Again, the ventricles, very odd shaped lateral ventricle, sort of a C shape, comes up and then comes back down, right? So two parts of the C there, and then the third ventricle, much smaller here in the center. Excuse me. Another view of the ventricles, highlighting these choroid plexuses, these capillary beds that are nestled within. And I've pretty much talked about cerebrospinal fluid again, that it derives from the plasma and flows through. And notice that it's produced by the choroid plexuses, but it's also produced by some other parts of the brain as well. So there are a certain special kind of glial cell that lines the ventricles called the ependyma cells. We didn't learn those, but they also produce some cerebrospinal fluid. So the lining of the ventricles produces cerebrospinal fluid. And notice a good portion of the cerebrospinal fluid is also produced in the subarachnoid space itself. So only some of the cerebrospinal fluid originates in the brain and passes through that pathway that we talked about. And again, it always is a modification or a filtrate of blood plasma. This describes the flow that I already talked to you about, getting down to the apertures in the fourth ventricle. And I want to get to this picture here to again show you eventually how the cerebrospinal fluid ultimately gets back to the sagittal sinus. So again, much of it is produced in the subarachnoid space itself, which is shown in light blue here. But this cerebrospinal fluid that was produced here in the choroid plexuses of the ventricles, as we said, it travels down the cerebral aqueduct into the fourth ventricle and then exits through the apertures into the subarachnoid space. And remember, we also have cerebrospinal fluid in the subarachnoid space of the spinal cord. Well, this cerebrospinal fluid in the subarachnoid space tends to get moved upwards just by pressure, because we're constantly draining fluid into this space. And so as fluid drains out of the apertures, it tends to push the fluid in front of it upward. And it follows the subarachnoid space up, 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 and eventually all gets pushed up to this median line right along the top center of the brain. So in dark blue here we see that sagittal sinus, right? That superior sagittal sinus that runs from front to back. And you'll notice right here we see some structures known as arachnoid villi or arachnoid granulations. These arachnoid villi look like little heads of cauliflower, basically. And notice what they do. They're a piece of the arachnoid mater, a highly folded piece of arachnoid mater that pokes through the dura mater, pokes through the meningeal layer of the dura mater, and extends into the sagittal sinus. And these little arachnoid villi consist of cells that can actively pump cerebrospinal fluid into the sagittal sinus. So this is the trick. This is how we get the cerebrospinal fluid ultimately back into the bloodstream, is through these arachnoid villi that poke into the sagittal sinus and pump cerebrospinal fluid from the subarachnoid space back into the bloodstream. So cerebrospinal fluid comes from the blood, and it eventually will flow and end up back in the blood. And again, as we said, this cerebrospinal fluid metabolically supports tissues with oxygen, nutrients, and so on. And again, by virtue of surrounding the cerebrum especially, provides buoyancy and cushioning. And the cerebrum is essentially floating in cerebrospinal fluid. All right, at this point, we now want to return to talking about specific structures of the brain. As I said earlier, it's organized a little bit differently. Your list of terms is organized from the cerebrum and works its way down to the medulla. But this presentation starts at the medulla and works its way up towards the cerebrum. So at this point we have to move and find on the bottom of the second page of the terms on this particular lab to Medulla Oblongata. As we said, this is the very bottom of the brainstem that's in communication or merges with the spinal cord. It's a slight enlargement. You can distinguish it from the spinal cords because it is slightly wider. It does flare out a little bit. And it begins right about at the level of the foramen magnum of the skull. So again, this little bit of enlargement here just above the spinal cord, that is the medulla oblongata. The medulla oblongata functions in a couple of things. Of course, it is the primary way that messages get to and from the brain, especially from the spinal cord. All messages from the spinal cord have to pass into the brain through the medulla. It does have several nuclei within it. Again, remember, nuclei are little islands of gray matter. Some of these nuclei are involved in cranial nerve origin. And so some of the cranial nerves that we'll talk about in the last week of the semester originate here in the medulla. As I said, all messages coming to and from the brain and the spinal cord pass through the medulla. It does have some nuclei, as I said. Some of the nuclei that you find here are involved in things like respiration, basic rate of breathing. You also have some nuclei here that are involved in blood pressure regulation. We also have some nuclei here that are also involved in heart rate regulation as well. The medulla is also part of what we call the reticular formation, which is a network of neuron cell bodies or nuclei extending throughout the brainstem, a loosely arranged network. And the reticular formation is involved in many, many basic autonomic functions. It also happens to be a part of the brain that's responsible for arousal, keeping the cerebrum essentially conscious. And so, as you know, a hard blow to the brain can shock the reticular formation, and this is what causes the loss of consciousness. Here's a cross-section of the medulla, and again, you can see that it has a variety of nuclei within it that are involved, again, in various body functions. All right, as we move up from the medulla, of course, then we encounter the pons, which, as I said earlier, is a large lump on the anterior brainstem just above the medulla. Once again, we see that the pons contain some origins of cranial nerves, different cranial nerves now from what we saw in the medulla. It is also part of that reticular formation that I just mentioned, which is involved in consciousness and sleep and so on. It also has some respiratory nuclei as well that work with the medulla to regulate respiration. And again, here we see the pons. At this point, we also have things referred to as cerebellar peduncles. A peduncle is a stalk-like structure. And so the cerebellum tends to connect to the brainstem through cerebellar peduncles. You can't see them on this particular image, but we will have an image later that will show you the cerebellar peduncles that attach to the brainstem here at the level of the pons. So I will highlight those later in another illustration. Another view of the pons on a real photograph of the brain. And then above the pons, of course, we have the midbrain. The midbrain is a very short segment of the brainstem that contains just a few things. The cerebral aqueduct that we mentioned, carrying cerebrospinal fluid from the third ventricle to the fourth ventricle, passes through the midbrain. We do have some nuclei in the midbrain that once again are part of the reticular formation. And once again, we do have some origins of a couple of cranial nerves. And again, we'll talk about all of the cranial nerves in another lab later in the semester. So there you see the very short little midbrain region. Little midbrain region here. We do have on the back side, on the posterior part of the midbrain, we do have a set of 4 bumps or bulges, and these are referred to as the corpora quadrigemina. The corpora quadrigemina, 4 bumps that are on the dorsal portion of the midbrain. If you break this word corpora quadrigemina down, corpora means body, as in corporal punishment, right, would be physical punishment of the body. So corpora means bodies, it's plural. Quadra means 4. Gemina is Gemini, that's twins. So it's 4 twinned bodies. In other words, 2 pairs of bumps. The upper pair of bumps are referred to as the superior colliculi, and then the 2 lower pair of bumps are called the inferior colliculi. Right? So 4 bodies, 4 bumps altogether, an upper 2 known as the superior colliculi, and a lower 2 called the inferior colliculi. These colliculi seem to be involved in relaying messages and certain kinds of reflexes. So the superior colliculi seem to be involved specifically in relaying visual messages and some visual reflexes like pupillary dilation and constriction. The lower pair seem to be involved more in auditory messages, relaying messages regarding sound to the brain, and also some reflexes associated with hearing, such as tensing of the eardrum in response to particularly loud sounds. They don't cause the reflex, but they're involved in the reflex or the relay pathway. And then a final thing that we see in this short region called the midbrain are another set of peduncles. Again, peduncles are stalk-like structures. I mentioned earlier that we have cerebellar peduncles connecting the cerebellum to the brainstem around the pons. Here in the midbrain, we have cerebral peduncles, and these are stalk-like structures that help hold the cerebrum in place on the brainstem. And so they serve a physical support to the cerebrum, but they also have tracts of white matter, again, passing through them. And so we do have signals passing up and down through the cerebral peduncles into the cerebrum. This is not the only way that the cerebrum attaches to the brainstem, and so it's not the only way that messages can pass up or down from the cerebrum to the brainstem, but it is a main way that messages can get to and from the cerebrum from the brainstem. As I said earlier, I was going to show you some pictures where we could see these peduncles a little bit better. This is a posterior view of the brainstem. I know this says ventral view, but that doesn't make any sense because you can only see the corpora quadrigemina from the... Oh, I'm sorry, this is a ventral view. Pardon me, this is a ventral view. So we see the pons and medulla, and you really can't see much of the midbrain, which is behind all this. But the reason I did include this is because what you can see are these stalk-like structures here, which are the cerebral peduncles. On the next view, which is the one I was... again, here's a view through the brainstem. Cerebellum. The cerebellum indeed is, as we said, the second largest part of the brain. 50% of the brain's neurons, about 100 billion neurons in the cerebellum. And the cerebellum has traditionally largely been associated with coordination of motor activity. So here's a picture of the cerebellum, again sitting behind the midbrain and the pons. You'll notice that, as I said earlier, the mostly gray matter, just a little bit of white matter, and that little bit of white matter forms sort of a tree-like formation. It almost looks like a tree trunk with branches. This pattern of the white matter is referred to as the arbor vitae. This tree-like pattern of the white matter is referred to as the arbor vitae, which in Latin means tree of life. Now, as I said, the cerebellum has usually been associated with coordination of motor activity. And so when we decide to contract our skeletal muscles, the initial conscious decision is made up in the cerebrum. But as those messages travel down towards the brainstem and eventually out the spinal cord to the muscles, those messages have to be modified and coordinated quite a bit. Some of the things that coordinate those signals and modify them are some of the nuclei that are up in the cerebrum itself, which we'll talk about a little bit later. But a lot of that coordination happens in the cerebellum. And so the messages pass into the cerebellum and get modified and then come back out through the cerebellar peduncles and continue on to the skeletal muscles. So it's a very important region of balance and coordination of muscle activity. Again, here we see the cerebellum on a photograph of the brain. And again, as I said earlier, just now, the motor coordination function of the cerebellum. Notice, though, that that's not its only function. And of course, being 50% of the brain's neurons, we should have suspected that it had more roles than just coordination of motor activity. And so you see there's quite a few different things now that we're learning about the cerebellum in addition to its motor function that these neurons are doing. So comparing texture of objects, how can I tell that something's smooth versus rough, for instance? Spatial awareness, perspective, recognizing, if I look at an object from the front and then look at it from the side, how do I know that that's the same object, and how can I connect that to the perspective that I had when I was looking at it from the front? Some basic rhythms, like keeping sense of how much time has passed as I'm performing tasks. Something to do with helping control eye movements. There's a lot going on with control of the eyeballs that we're not really aware of, but just a simple thing like turning your head and maintaining your gaze, say, on something like a flying bird, takes an incredible amount of coordination to keep your eyeballs focused to get a nice stable image. Cerebellum seems to be involved in that as well. The cerebellum seems to play a role in evaluating the pitch of sounds that we hear, which is partly involved in distinguishing between certain words and voices, recognizing individual voices. And also some level of planning and scheduling, which is a big surprise because we usually associate that with the cerebrum. But the cerebellum does seem to work with the cerebrum in doing that, and some level of emotional control. Now, at this point, I want to make a side comment. All of these functions that we're talking about in the brain are highly generalized. We're being very superficial in our treatment of the brain. The brain's an incredibly complicated organ, and there's very few parts of the brain that actually work solely on one task in complete isolation of any other part of the brain. And so when I said down here, when it says planning and scheduling, well, it doesn't do it all by itself. It works with the cerebrum and other parts of the brain to perform that function. And many, many parts of the brain work hand in hand, one with another. So as we go through this entire lab telling you what different parts of the brain do, something you want to keep in the back of your mind at all times is that, well, that may be a main function, or it may play a large role in that function, but it's almost never the only part of the brain that's working on that function. The brain is an integrated whole, and it needs all of its parts to perform all of its functions. A lot of what we've learned about brain function has unfortunately been derived from studies on victims of brain injury. So a certain part of the brain gets injured in an accident, and then we assess to see what function has been lost. And so that's where a lot of this has come from. But just because we lose function when one part of the brain is damaged doesn't mean that that part of the brain was solely responsible for that function. It could have been working, and it does work with other parts of the brain in tandem to perform that function. So much of these functions are integrated throughout many parts of the brain. I think this has also led to some misconception about how much of the brain we use. Oftentimes, you will see that if you have minor brain damage due to accident or injury, you can recover function because other parts of the brain will take over and replace function that's been lost in one small part of the brain. Again, this highlights the integrated nature of brain function. Well, because you can have another part of the brain kick in and take over the function of a lost part of the brain, a small part, that led to the mistaken notion that we only use 10% of our brain or 30% of our brain. This idea that we don't use all of our brain and we're keeping some of it in reserve in case of damage. Well, that's just not true. We are using our entire brains all the time. There's just a lot of redundancy, again, and overlap in function. And so just because another part of the brain can help take over some lost function doesn't mean that that part of the brain wasn't doing anything previous to the injury. Okay, as we move up from the cerebrum and the midbrain, we then enter a region, fairly complicated region of the brain called the Diencephalon. The Diencephalon, you'll notice, is this upper part, this blue part here, that we were just just above the midbrain, which we were just talking about. So all of this is the Diencephalon. You'll notice in some people's way of thinking, the Diencephalon is part of the forebrain, what people call the forebrain. I don't particularly care for these terms, forebrain, midbrain, hindbrain. It just gets very confusing, and I also am not a big fan of the encephalon terms. These encephalon terms are really more appropriate for embryonic structure. These are actually embryological regions of the brain, and then we carry them out to the adult structures that they develop into. But again, it gets fairly confusing because there's this sort of overlap between forebrain, midbrain, metencephalon, myelencephalon, and so on. And so I tend to just stick to the basics, medulla, pons, midbrain, diencephalon. There's really not a good term for diencephalon. I've heard some people refer to it as the interbrain, perhaps, but generally most people refer to this just as the diencephalon. And then cerebrum. All right, cerebrum, which again they want to call telencephalon, which I don't care for. But at any rate, the cerebrum and the diencephalon, if you like that kind of organization, are part of what's called the forebrain. Anyway, the diencephalon, as I said, relatively complicated region, lots of interesting parts here in the diencephalon. Basically, it consists of three things: the thalamus, the hypothalamus, and the epithalamus. And I do want to remind you that located right in the middle of the diencephalon is the third ventricle. So right in the center of this region, diencephalon, you have a hollow fluid-filled chamber, the third ventricle. So keep that in mind as we talk about the parts of the diencephalon. The major part of the diencephalon is the thalamus. Notice about 80% of the diencephalon is the thalamus. The thalamus consists of two masses of gray matter. So in essence, it's a kind of nucleus, two masses of gray matter that are on either side of the third ventricle. So the two halves of the thalamus are separated by the third ventricle. Now, they aren't completely separated. There is a tract of tissue that runs across the third ventricle and connects the two halves of the thalami, and this is known as the intermediate mass of the thalamus. It's a little tract of nerve tissue that runs through the third ventricle from left side to right side. And again, the thalamus is a fairly complicated structure consisting of, again, nuclei, gray matter, in each of the halves. Now, the role that the thalamus plays is primarily as a gateway to the cerebral cortex. And what we mean by that is that, as we saw with the cerebral peduncles, you have to be able to get messages up into the cerebrum from the brainstem. So we saw cerebral peduncles do that, but notice the vast majority of messages that are traveling from the brainstem up into the cerebrum pass through the thalamus. So the cerebral peduncles, while they allow some of the messages to go up, they serve mostly as physical support for the cerebrum. The vast majority of messages pass through the thalamus. And so the thalamus is like a routing station, a relay station. And so the signals come in to the thalamus, and then the thalamus makes connections that decide where to send these signals. So remember, I mentioned earlier that the postcentral gyrus was an area of sensation, skin sensation, touch. Well, we have messages, signals of touch coming in from all over the body. How do we know to funnel all those signals coming in from all these disparate parts of the body up specifically to that postcentral gyrus? Well, that's the job of the thalamus again, is the routing station. It's responsible for receiving all those sensory signals regarding touch and routing them in a particular pathway that takes them ultimately to the postcentral gyrus. That's just one example of how the thalamus is going to route signals to their correct destination in the cerebrum. So that's its main function. In addition to that, it also is involved in some basic memory and emotion, because it's part of a system known as the limbic system. We don't have the limbic system on our list of terms, just like we didn't have the reticular formation. Excuse me. But the limbic system is basically a set of structures that are above and beside the thalamus that are involved in memory and emotions, and basically sort of part of our animal brain. Conversion of short-term memory to long-term memory happens in the limbic system, specifically a part of the limbic system called the hippocampus, which is not on our list of terms. And then some of our most basic emotions are mediated or have something to do with the limbic system. And so the thalamus, at least parts of the thalamus, are part of the limbic system. And so they are also involved in some of these memory storage and emotional functions. Some people have argued also that the thalamus is involved in some level of initial or dim awareness. Not so much conscious awareness, but sort of reflexive, subconscious awareness of things. The example that was taught to me was, let's say you're walking through a forest, and out of the corner of your eye you see a stick. Well, it's only in your peripheral vision. It hasn't even consciously registered that it's a broken branch or stick yet. But you get startled because somewhere in your subconscious it might have been a snake. Because after all, you're in the forest and it's possible that there is a snake out there. And then, of course, once you turn your head and focus on it and consciously registers as a stick, that initial reaction subsides. But that initial subconscious, dim, kind of reflexive awareness of something is also the kind of function that might be played by the thalamus. Here again we see the central location of the thalamus, again on either side of the third ventricle of the brain. Thalamus again through different views. All right, a second part of the diencephalon is the hypothalamus. The hypothalamus essentially is located just beneath the thalamus. And so it forms the lower walls and the floor of the third ventricle, whereas the thalamus, again, formed the sides of the third ventricle. The thalamus has a couple of lumps on the bottom of it, referred to as the mammillary bodies. The mammillary bodies, again, these two bumps on the bottom of the hypothalamus, are involved once again in the limbic system and seem to be involved again in specifically memory, the retention of memories. It also has a stalk-like structure coming off the bottom of it called the infundibulum. The infundibulum is a connection between the hypothalamus and a gland below the hypothalamus known as the pituitary gland. And the pituitary gland is something we talk about when we do the endocrine system in the AMP2. Just for now, know that the pituitary gland is a very important gland, produces hormones that help regulate other glands of the body. And it also releases a couple of hormones that are actually made by the hypothalamus itself. Right, so the hypothalamus sits below the third ventricle largely, has two bumps on the bottom called mammillary bodies involved in memories, and stalk-like infundibulum that extends down to the pituitary gland. And again, the pituitary sits, just to give you a sense of orientation, sits in the sella turcica of the sphenoid bone in the skull. So remember, in the floor of the cranium, you had this Turkish saddle, this depression in the floor of the skull. That depression is where the pituitary gland sits, very well protected by the bony crests of the sella turcica. Again, in terms of location, excuse me, if this was the thalamus in this region, the hypothalamus is just below it. So all this region that funnels down, and then on the bottom of the hypothalamus you have a lump here. That's the mammillary body. Again, this is the sagittal section, so there'd be a second lump on the left side, so two bumps. And then a stalk-like infundibulum that leads to the pituitary gland, which sits in the sella turcica. The hypothalamus is an incredibly important part of the brain, even though it's not terribly large. It's an incredibly important control center for the autonomic nervous system and also for the endocrine system. In terms of its endocrine function, it does release several what we call releasing and inhibiting hormones that control pituitary function. And so while some people refer to the pituitary gland as the master gland of the body, I actually think of the hypothalamus as the master of the endocrine system, because it is the master of the pituitary, which then goes on to affect other hormones and glands in the body. So it really is a major control center for a lot of endocrine function. Not all, but a lot of endocrine function. The hypothalamus itself also produces two hormones that are released from the posterior pituitary gland, and those hormones are oxytocin and antidiuretic hormone. Oxytocin is involved in labor contractions and lactation. The synthetic version of oxytocin is called pitocin, and many women have received pitocin to encourage labor and delivery. Antidiuretic hormone is a very important hormone that affects water conservation by the kidneys, and by affecting how much urine we produce, that actually ultimately is an important hormone in regulating blood pressure. Again, these two hormones, oxytocin and antidiuretic hormone, which some people call vasopressin, these are actually made by the neurons of the hypothalamus. And those neurons will send axons down through the infundibulum, where the axons terminate in the posterior pituitary gland, and release these two hormones. And so what we're seeing here is the very close relationship between the nervous system and the endocrine system. These two hormones that I just talked about are actually secreted by neurons from axon tips. Normally, we would call that kind of chemical a neurotransmitter that comes out of the tips of an axon. But in this case, they're not crossing a synapse and activating a second neuron. In this case, they're being released directly into the bloodstream. And that really is the hallmark of a hormone, is a regulatory chemical that's released into the blood for distribution. And so we see neurons functioning as hormone-producing cells. So that hormonal function is a very important part of what the hypothalamus does. Hypothalamus, as I said, is also involved in the autonomic nervous system. A lot of decisions about what kind of response, sympathetic or parasympathetic responses, are made here in the hypothalamus. And those will ultimately affect autonomic messages that affect heart rate, blood pressure, digestive system activity, etc. In addition to those two basic controlling functions, the hypothalamus also serves many sort of just basic housekeeping functions like thermoregulation. It is the body's thermostat, sensing body temperature and creating reflexes that respond to it, such as sweating. It is your hunger center, and basically the part of your brain that tells you when you're full in response to blood chemistry conditions in the stretch of the stomach. It also is your thirst center. It tells you when you're thirsty. And again, this closely tracks the concentration or osmolarity of the blood. And this is also how we affect blood pressure, because as you get dehydrated, your blood pressure falls. You get thirsty, you take in water, and you produce more antidiuretic hormone to reduce water loss at the kidneys, and this helps increase blood volume, which then increases blood pressure. So our sensation of thirst isn't important just for keeping us hydrated, but it also is important in blood pressure regulation again. It seems to work with some other parts of the brain in generating sleep patterns and biological rhythms, annual or daily rhythms. Again, the mammillary nuclei of the hypothalamus are involved in memory, working with the hippocampus, which is that part of the limbic system that is involved in converting short-term to long-term memories. And it is finally also a basic emotional center for the body, so it's a basic seat of anger and aggression. Fear seems to be modulated through the hypothalamus. Some basic sensations of pleasure and contentment and satisfaction, both psychological and physical, and it is also the seat of sex drive. So that's a lot of different basic functions being carried out, all essentially automatically for us by this very important region known as the hypothalamus. And here we see the hypothalamus, and again the variety of nuclei that it contains in order to perform all these different functions that we just talked about. A view of the location of the hypothalamus on a photograph. And finally, we move to the last part of the diencephalon, which is the epithalamus. The epithalamus is a very small portion of the brain that is located essentially posterior to the thalamus, just behind the thalamus. Again, it's got portions of it that are involved in the limbic system, especially important in sending messages to and from the thalamus regarding limbic types of basic emotions and awareness and activity. But perhaps the most noticeable portion of the epithalamus is a structure called the pineal gland. It is a gland of the endocrine system, but it's also part of the brain, and is shaped like a pine cone, and thereby gets the name pineal gland. The epithalamus again is located just behind the thalamus, so this region, this very small region right here, is what we call the epithalamus. And again, the most noticeable part is a single pine cone shaped gland right here, just above the corpora quadrigemina is the pineal gland. Here we see a photograph. The pineal gland, before we move on to the cerebrum, the pineal gland seems to be work with the hypothalamus in basic rhythms. These are both daily rhythms, like our sleep cycle, perhaps monthly rhythms like the estrous cycle or menstrual cycle in women, and even annual rhythms. So some things that are associated with the pineal gland, for instance, would be jet lag, Why do we get jet lag if we travel long distances over short periods of time? Again, because we have a daily rhythm. It's called a circadian rhythm of activity, and that generally is close to a 24-hour rhythm. It's not a perfect 24-hour rhythm. Fortunately, it gets reset by sunlight. But for a few days after we travel, and our clock is a little off from local time, we just sort of feel out of sorts. Something's not quite right, because our daily rhythm is not in synchrony with the local time. And it does take a few days for the new sunlight pattern to reset our circadian rhythm. And this is largely mediated through the hypothalamus and the pineal gland. The pineal gland secretes a hormone called melatonin, which seems to be involved in these rhythms, and that's why, not surprisingly, you see melatonin on the shelf as a supplement that might aid in people that have difficulty sleeping. Whether it works or not, I'm not sure. I tried to take it once myself, and it just made me dizzy, so I stopped taking it. But for some people, they say it works. It's also involved in annual, as I said, rhythms. These are called circannual rhythms as opposed to circadian rhythms. Annual rhythms have to do with things like your mood, and again, pretty much track sunlight and how long the days are. We know that days get longer, of course, in the summer and shorter in the winter. Some people suffer from what's called seasonal affective disorder, SAD, where they get depressed in the wintertime. And that has to do with the shorter days and how that loss of sunlight affects the pineal gland and melatonin production, and it affects your mood. And in some cases, some people get severely depressed during the winter time. Some people who move to very high latitudes, like Alaska, for say a new job, discover that they can't handle it because the days are much shorter in Alaska across the board, and they just find that they get very depressed when moving to a place like Alaska and can't remain there. These are all very real conditions and largely mediated through melatonin and the pineal gland. All right, and so finally then on to the cerebrum, which is back to where we started. When we looked at surface anatomy of the brain, we were looking at the cerebrum. So now we're back, and as I said earlier, we're going to take a quick look at the lobes of the cerebrum and talk about, again, very generally, what kinds of functions take place in the cerebral lobes. So again, here's the cerebrum, and the lobes of the cerebrum are the frontal lobe, which essentially is all of the cerebrum in front of the central sulcus. The parietal lobe, which is behind the central sulcus. So again, here's the central sulcus. So this would all be frontal lobe. This would be parietal lobe right here. Back in this region, we have what's called the occipital lobe, and you'll notice these regions are named for the cranial bones under which they lie. This portion right here is, of course, the temporal lobe underneath the temporal bone. And those really constitute the four major lobes of the cerebrum. There is a deep lobe underneath the temporal lobe. If we pulled apart this lateral fissure and looked inside there, we would see another fifth lobe of the brain called the insula. But you cannot see that from the surface, but we'll see a picture of it in just a little bit. So each lobe generally has some very characteristic functions associated with it. And again, this is very complex. We're just going to try to very briefly summarize what happens. The frontal lobe is generally referred to as your seat of consciousness. So this is where you do voluntary muscle contractions, planning, analysis, conscious memory, emotion, origins of aggression, conscious interpretation of events that then trigger aggression, which then is mediated, of course, through the hypothalamus. Again, many parts of the brain work together. But this is sort of you. your personality, the thing that makes you you at a conscious level, that's pretty much localized in the frontal lobe. The parietal lobe is primarily a sensory lobe. And as I said earlier, of course, it general senses like touch and temperature and things like that, but also some taste and some visual information. The occipital lobe is also primarily a sensory lobe, and it is almost exclusively dedicated to vision. It is primarily where you consciously interpret visual signals coming from your eyeballs. The temporal lobe also is primarily a sensory lobe again, and it is involved in a variety of senses. Hearing seems to be localized here. Smell seems to be largely localized here. It is involved with memory, as really all the lobes are, because to have memory, you remember things you felt, you remember things you saw, you remember things you heard, and they all have to work together to create a memory. And then again, the insula, which is the deepest one, hidden pretty much under the lateral fissure, seems to be more involved again in putting things together, in integration of information, especially language and vision, putting signals together to make sense out of language and vision, and does serve a little bit of a taste function as well. So lots of overlap between these lobes, who are just being very general in these functions that we're listing for these parts of the cerebrum. Here you see a nice diagram of the location of the lobes again, and in this particular diagram, again, they've separated the lateral sulcus, I called it fissure earlier, but the lateral sulcus to peek underneath and see the insula, which is underneath there. And then a nice sort of tabular version of the different functions of these lobes. And then in photographic images, the frontal lobe in front of the central sulcus. And remember I said that precentral gyrus that sits just in front of the central sulcus was largely devoted to motor signals, sending messages out to skeletal muscles of the body. And one nice thing that is fairly common to do is to draw a map on the surface of this gyrus indicating which parts of the gyrus and how much of the gyrus are devoted to particular muscles of the body. And this little map, this little figure, is referred to as a homunculus. And it is nice, it's a nice visual representation of how much of our brain is devoted to moving muscles in particular parts of our body. So you notice, for instance, we don't have a lot of neurons devoted to the trunk, for instance, because we don't have a lot of motor units there. We tend to have larger, more powerful motor units, and that doesn't take as much innervation. But in the hands and the face, of course, we have lots of fine muscle contractions, and so the muscles there tend to be constructed of smaller motor units, and so we need more neurons to control those very fine muscles of facial expression and manual manipulation. And so this homunculus is a nice representation reminding us of how much more precise and how much more control we have over muscles in these areas. Again, the parietal lobe sits behind the central sulcus, and as we just did with the precentral gyrus, we can also draw a homunculus on the postcentral gyrus, but now this one is a sensory homunculus, showing how many sensory neurons we have devoted to various parts of the body. And once again, you see the predominance of the hands and the face in terms of some very sensitive parts of the body. Occipital lobe again in a photograph. Temporal lobe, just where you expect it to be. And then the insula again, sitting deep. But this gives you a nice sense of where that lobe is located underneath the temporal lobe. The temporal lobe here and the parietal lobe up here. All right, so again, we've sort of summarized the functions of each lobe. This I just included to show you that even within lobes themselves, there's various parts that are devoted to certain things. So in the In the frontal lobe, for instance, there's a little patch called Broca's area that is involved in language, for instance. Notice here on the parietal lobe, there's a little patch that's associated with taste. So even within a single lobe, the functions can be somewhat localized in different regions. I don't expect you to memorize any of this. I just wanted to highlight that even within a single lobe, there can be localization of some functions. Language is a very interesting and complicated subject. We're not responsible for this. I just find it interesting, so I threw it in at the end of the video. I mentioned Broca's area, just highlighted Broca's area on the frontal lobe. Broca's area is the area that actually generates the motor signals that travel to the muscles of your larynx, your tongue, cheeks, lips, everything for speaking. All the motor signals that are involved in both verbal and nonverbal communication come from Broca's areas. Wernicke's area is farther back in the brain, back by the parietal lobe, and it is involved in the planning of speech. So it formulates the phrases in your head, the plan of what we're going to say. Then it sends that plan to Broca's area, and Broca's area is what actually sends out the signals to the muscles. And those tend to be located on only the left side of your brain, interestingly. And so here we see again Broca's area in the frontal lobe, and then Wernicke's area back on the parietal, sort of parietal temporal lobe area. These are involved in language again. Aphasia is loss of language ability due to some damage to these areas. If you damage Broca's area, you can think of what you want to say just fine. You just have trouble saying it. Because again, Broca's area is what sends the actual messages to the muscles. And so in your head, you know exactly what you want to say, but you have difficulty in actually saying the words, saying them in the correct order and things like that. If you have damage to Wernicke's area, you have what's called fluent aphasia, where it is very difficult for you to actually formulate any kind of coherent speech. So you can send messages out to your muscles just fine, and so you can say words normally, and in fact these people tend to talk quite a bit, but they don't say anything sensible because they can't formulate a plan of what they want to say because Wernicke's area has been damaged. And these people also tend to have difficulty in comprehending speech as well. As I mentioned earlier, the two halves of the cerebrum are also not mirror images of each other. They generally provide or perform similar functions, but there are nuances of difference. And so this figure shows the what we call the lateralization of cerebral functions. One thing to notice, first of all, is that in some cases the two halves of the brain are doing the same thing. So in this patch right here on the precentral gyrus, you have control of the hand, and in the same area you have control of the hand. But notice what's different is which hand. Right hand control here, left hand control here. And what you'll clearly immediately notice that control of the right hand is performed by the left precentral gyrus, and control of the left hand is performed by the right precentral gyrus. This is because many of the fibers that we send out to the body undergo what's called decussation in the medulla. Decussation means crossing over. The fibers cross over to the other side. And this largely happens in the medulla. Not all motor fibers do this, but many do. So not surprising to find that the one half of the brain controls and senses things on the other side. It also works with sensation as well, right? So feeling with the right hand is on the left side of the brain, and so on. The other thing I want to point out here is that, again, the complementarity of the two parts of the brain. And so in similar areas, so on the left side we might have verbal memory of words, but on this side we have memory of shapes in the same area. So the two halves are doing something similar. They're both involved in memory, but not necessarily memory of the same exact thing. As I said, on the left side, you have Broca's area involved in speech. In the same area on the right side, you have an area that's largely devoted to language comprehension, understanding spoken language. So again, related but not identical. This is what we mean by the complementarity of the two halves of the cerebrum. Back here we have rational symbolic thought, logical thought. In the same place on the right side, intuitive nonverbal thought, feeling, and so on. And so this is, again, all known as lateralization of the cerebral function. The left hemisphere, excuse me, the left hemisphere is generally what we call the categorical hemisphere. So what we mean by that is it's the analytical side of the brain for sequential thinking, analysis, planning, breaking down information into bits and then putting it back together for understanding. And again, spoken language, written language. It's sort of the logical, cognitive, I don't want to say cognitive, just the rational side of the brain. The right hemisphere, on the other hand, tends to be what we call the representational hemisphere. And so instead of perceiving information in pieces that get put together in a reductionist sort of fashion, it tends to interpret or understand things in a more integrated, holistic way. And so higher cognitive functions like imagination and insight would be on the right side, whereas analysis is being done on the left side. Musical skill, artistic skill, athletic coordination, and creativity located on the right side of the brain. The perception of patterns located on the right side of the brain. And then comparisons between all your senses. Again, very holistic kinds of functions. There's an unfortunate tendency to polarize this or dichotomize this and talk about people as being right-brained or left-brained. So scientists we think of as maybe left-brained individuals where the left hemisphere predominates, and artists, performers are right-brained individuals. I find that to be unfortunate because it really downplays the requirement of both sides of the brain for both kinds of activities. So for instance, when you think of a scientist, one of the most important things scientists have to be able to do is perceive and recognize patterns. Especially biologists have to perceive patterns in nature. And so this side of the brain has to be very well developed to be a successful scientist, but that's on the right side. And then, of course, to be a good performer, you have to be very able to, for instance, memorize your lines if you're an actor. And if you're an artist, it certainly helps to be able to know about the components of a piece of art that you're looking at and to be able to analyze it, or to even think about the art that you're about to create from this logical sense. All artists have a goal in mind, or usually have a goal in mind when they create a piece of art. So, excuse me, it's really unfair to think that people with different aptitudes or skills are using one side of the brain more than the other. Interestingly enough, whether you're right-handed or left-handed has something to do with this lateralization, but it's not a perfect alignment. It's not a perfect correlation. So in right-handed people, for instance, which is the more common condition, the left hemisphere is the categorical one, as we just described, in about 96% of right-handed people. In a few cases, the hemispheres are reversed, and the right hemisphere is categorical, and the left hemisphere carries out more of the pattern type recognition. But notice it's a small percent in right-handed people. But in left-handed people, the left hemisphere is the categorical one, as we described, in only 70%. So still, most people are, the left hemisphere is categorical, but not nearly the same percentage as in right-handed people. So a much higher percentage of left-handed people have the right hemisphere being categorical. Even more interesting to me, in 15% of left-handed people, neither hemisphere is specialized. Both hemispheres serve both functions. So even with this lateralization, we see that there's variation among individuals, and left-handed people tend to be more variable in this lateralization than right-handed people do. Also, there are sexual differences. Males tend to be more lateralized than females, and children tend to be less lateralized. And as we get older, we tend to become more lateralized. Again, just a view of the gray matter and white matter to finish this out. I think at this point we've pretty much covered every term that's on our list of terms. I don't believe we've missed anything, and so with that, I will just direct your attention to the second video for this week. The second video is going to be models of the brain that we have in our labs, and I'm going to point out every item that I can on the models that we have, and that won't take nearly as long as this introduction to the brain did. So until the next video, take care, and I'll see you soon.